I'm sure you all know this molecule. Yes, it's DNA. You always wanted to know about it since you have heard about it for the first time, isn't it? So let's go ahead to know about it. Namaste. We've learnt about chromatin in previous classes. Chromatin is made up of DNA and histones. DNA contains all the information that is required for the growth, reproduction, and to pass the hereditary characters to the next generation. The structure of DNA was discovered by James Watson and Francis Crick in 1953. DNA, as we all know, is deoxyribonucleic acid. It's a macromolecule found in our cells. DNA is a polymer of nucleotides. To understand what a polymer is, let's take an example of a compound that we are familiar with. This is starch. Yes, starch is a polymer of glucose, which means many glucose molecules join together to form a starch. So, can we say that DNA is also a polynucleotide chain? Yes, we can. So, what is DNA made up of? DNA is made up of two strands which are twisted spirally as a double helix. These two strands are held together by hydrogen bonds and are complementary to each other. So what does the word complementary mean? Complementary means combining in such a way that they are compatible to each other or they support each other. Now, one complete turn of DNA consists of 10 nucleotides and is of 34 angstrom. 1 angstrom is 10 raised to minus 7 millimeters. So it's 34 angstrom long and 23 angstrom wide. DNA is made up of many nucleotides. So what is each nucleotide made up of? We need 3 units to build up our DNA: deoxyribose sugar, nitrogen base pair, and phosphate group. Deoxyribose sugar is a pentose sugar, is a 5-carbon sugar molecule. DNA gets its name from deoxyribose sugar molecule, that is deoxyribonucleic acid. Another important unit of DNA is nitrogen base pairs. There are 4 nitrogen base pairs in a DNA molecule. This consists of adenine, thymine, guanine, and cytosine. It's quite hard to believe that whole our life is written with the help of these 4 letters. Adenine and guanine are purines, and thymine and cytosine are pyrimidines. It's boring when we don't remember or get confused with these terms. So here's how you can remember them. You can remember them with the help of this mnemonic device. Purines are pure as gold. Purines are pure as gold. So pure, P-U-R, is for purine, and as gold, A-G, adenine and guanine. And for pyrimidine, cut the pie. So CUT cut will help you to remember C for cytosine, U for uracil, which is found in RNA, and T is thymine. Cut the pie, so PY is for pyrimidine. Yeah, you can remember it like this. Okay, let's go ahead. So the sugar and the phosphate group forms a sugar phosphate backbone in a DNA. These two forms 2 strands of the DNA. So we now know that DNA is a double-stranded structure. These are the 2 strands. these strands will run parallel but in an opposite direction. This means they are anti-parallel to each other. Nitrogen bases, they bind to each other as adenine binds to thymine, and they have a double bond between them. This double bond is a weak hydrogen bond. Okay. You have to show this weak hydrogen bond by a dotted line, okay, which will represent it's a weak hydrogen bond, right? So I'll present nitrogen base pair as A double bond T. Can we also write T double bond A as well? Yes, we can. The mnemonic device to remember A binds to T is ants are on the tree. So A binds to T. Yes. And guanosine binds to cytosine with a triple bond. Again, this one is a weak hydrogen bond. So we can represent this as G triple bond C. Or we can write this as C triple bond G. The mnemonic device to remember G and C bonding is gorillas of the Congo. So you say G binds to C, gorillas Congo. Okay, or you can say camels of the Gobi Desert. Okay, so C binds to G, camels and Gobi. Right? Okay, let's go ahead. Let's do a quick check of random sequence in our DNA to find out if you have understood that. Okay, I'll give you a sequence and you will give me a complementary pair. Right? So here goes the sequence ATTGC What is the correct complementary sequence to that? What do they bind to? The correct complementary sequence is AT, TA, GC, CG, TA, GC and CA. Hope you have got this one right. Now, as per the Chargaff's rule, the total number of purines and pyrimidines is always equal. That is, A plus G is equal to T plus C. Okay, that is, the total number of adenines and guanines will be always equal to the total number of thymine and cytosine. Right? Okay. A human genome contains 3 billion base pairs of DNA packed into our 23 chromosomes. That is 6 billion base pairs of DNA packed into our somatic cells. So how many base pairs do we have in our chromosome? A human chromosome number 1, which is the largest, has 249 million base pairs in it. Quite interesting, isn't it? Okay, this molecule appears like a DNA, but it's not. Can you spot the difference? These are a few differences. RNA is a single-stranded molecule. RNA contains ribose sugar in place of the deoxyribose sugar in the DNA. And the third one is that they have uracil in place of the thymine. Hope you have enjoyed learning this biomolecule. See you in the next. Thanks for watching.